Hi, today we'll be looking at microbial limit testing, or MLT, which is a key microbiological quality control test that is used in pharmaceutical and personal care industries. Now, this session will provide an overview of what an MLT is, why it is performed, and how it is practically implemented in order to meet regulatory and product safety standards. Now, a microbial limit test refers to the compendial microbiological assay that's required in order to ensure that a product's microbial quality is within generally acceptable limits. And this applies to a broad range of products, including pharmaceuticals, cosmetics, personal care formulations, and their active pharmaceutical ingredients, also known as APIs. And the objective of this entire process is to ensure and detect and quantify any microbial presence and confirm the absence of certain harmful, objectionable organisms. Now, MLT applies to non-sterile pharmaceuticals, raw materials, or APIs, and finished products. And the purpose of MLT is not necessarily to confirm sterility. That is a domain of sterility testing. But the purpose of MLT is to rather ensure that the microbial levels are controlled and safe for intended use. And the testing is critical in quality assurance pipelines where the products are just not terminally sterilized. And the methodologies for MLT are established by international pharmacopoeias, including the United States Pharmacopoeia, the European Pharmacopoeia, and also the Japanese Pharmacopoeia. Now, all compendial approaches generally share one common goal, and this is to ensure microbial quality complies within the good manufacturing practices, also known as GMP. Now, there are two main goals of MLT, and this is first to assess the total microbial load that's present in the product. And this microbial load includes aerobic bacteria, yeast, molds, and other types of organisms. But second, it's to screen for the presence of certain objectionable organisms or pathogens that should never be present depending on the product type and its administration route. Now, why is the MLT process so important? Well, it's important for so many reasons, but to start off with, it's important because it safeguards patient safety. Now, microbial contamination, even at just seemingly extremely low levels, can definitely cause infections in vulnerable populations and populations like neonates or even just immunocompromised patients. But second is that it also supports compliance, regulatory compliance with pharmacopoeial and also GMP standards, and it's just generally just a part of the processes that companies need to go through in order to get their products approved. But third is that it also preserves a sense of product integrity. Now, contamination can lead to product spoilage and physical changes, and definitely lead to a shorter shelf life of a certain product. But finally, the MLT also mitigates the business and legal risks that may be involved in having a product passed. Now, a failed test in the MLT can definitely result in recalls, regulatory action, or even brand damage of certain companies that propose certain products. So definitely an MLT is required in order to show that a company's product complies with regulatory and safety standards. Now, there are many types of MLT, and MLT can include both quantitative and qualitative testing approaches. Now, under quantitative tests, we generally measure the total aerobic microbial count, also known as TAMC, and we also measure the total yeast and mold count, also known as TYMC. And under qualitative tests, we generally look for very specific objectionable organisms, which may include the following here. And acceptable limits and target organisms may vary by product type, such as even oral versus topical formulations, and they're generally based on pharmacopoeial guidelines. Now then, how do we prepare a sample and get it ready for an MLT test? Well, correct sample preparation is essential for accurate MLT tests, and this entire process of having to prepare a certain sample to use for the MLT involves the following. First, starting with dilution. Now, we generally try to reduce the concentration and eliminate the inhibitory effect of the excipients or even the preservatives, so we dilute it down. But second, we also neutralize it. And the purpose of this neutralization stage is in order to inactivate any antimicrobial agents and also prevent any false negatives. Third, we go into a process of homogenization, and this entire process is in order to ensure a generally sort of even microbial distribution across the MLT testing platform. And fourth, we use controls, and this includes both positive and negative controls in order to validate method performance. And at the end of the day, aseptic handling is definitely required in order to avoid cross-contamination, and aseptic handling is required throughout the entire process of doing the MLT. And alright, that is the end of this video, and thank you for watching, and in our next video, we'll be looking at the various testing methods for MLT.